Hello everyone. Today our topic is mechanism of DNA replication, which is an extremely enzyme oriented reaction. Okay. So DNA replication meaning the production of new DNA molecule from the old DNA or parent DNA molecule. Yes, which are exactly similar to the parent DNA, that is known as the DNA replication. And DNA replication process is extremely rapid, okay, and it requires many enzymes and energy, which is called ATP. So I have told you already that mechanism of DNA replication is enzyme oriented, and this mechanism is extremely rapid, okay. So now you should know the DNA replication process begins or occurs during the S phase of interphase in cell division or in cell cycle. Okay, so it occurs, the DNA replication occurs during the S phase. S means what? S means synthetic phase. Okay, so DNA replication occurs during the S phase of interphase of cell cycle or cell division. So here say these are the important enzymes involved in the process of DNA replication in prokaryotes. Okay, remember this is very important. The name of the enzymes are very important. So these are the complete set of enzymes which are involved in the mechanism of DNA replication in prokaryotes. So the first one is here say that DNA polymerase 1. So in prokaryotic DNA replication, the DNA polymerase 1 removes the RNA primer and replaces it with newly synthesized DNA. So I will tell you what is RNA primer. Okay, simply remember DNA polymerase 1 removes the RNA primer, and in those places it synthesizes new DNA strand. Okay, now come to the second one, which is DNA polymerase 2. So DNA polymerase 2 enzyme has the repairing function. Okay, it can repair the mismatched pair. Okay, I will tell you later. So now the third one, see DNA polymerase 3. So DNA polymerase 3 is the main enzyme in prokaryotic replication because it synthesizes the new DNA strand always into the 5 prime to 3 prime direction. That means the DNA polymerase 3 always synthesizes a new strand which is in the 5 prime to 3 prime direction. Okay, always new strand is synthesized from 5 prime to 3 prime direction. Now the next one, see the next one is the helicase enzyme, again very important enzyme. So helicase enzyme helps in the breakdown of hydrogen bond present between the two strands of DNA. Okay, so helicase enzyme breaks the hydrogen bond present between the two strands of DNA or present between the nitrogenous bases. Next one is the ligase. So ligase helps in filling up the gaps between the Okazaki fragments. Okazaki fragments are very short fragments of DNA. Okay, so it seals the gaps between the Okazaki fragments. That means the fragments of DNA to produce a continuous DNA strand. Okay, so that means the ligase functions as sealing enzyme. Okay, that means which seal the gap or which fill up the gaps between the Okazaki fragments. Now the next one is the primase. What is the function of primase? So primase enzyme helps in the production of or synthesis of RNA primer. Okay, so RNA primer is very important. So RNA primer is a short strand of RNA which is very important for the synthesis of the new DNA strand. Okay, next one is the sliding clamp, and sliding clamp helps to hold the DNA polymerase. So sliding clamp helps to hold the DNA polymerase in place when nucleotides are being added. That means DNA polymerase 3 enzyme, this one third number, the DNA polymerase 3 enzyme polymerizes the nucleotide in a long chain. Yes, and during this process, the sliding clamp hold the DNA polymerase enzyme in place in the DNA strand. Okay, so now the next one is the topoisomerase. So topoisomerase enzyme relieves the stress on DNA, or you can say it relieves the tension in DNA. So it unwinds the breaks and then reseals the DNA. That means I will tell you the function of topoisomerase. Yes, see in the next slide. So enzyme topoisomerase attaches to the two forks of bubble to relieve stress on the DNA molecule as it separates. So this is the structure. So here see, this is the enzyme. The yellow color is the enzyme. Yes, this is the enzyme. So this enzyme binds to to this part. Okay, this is called the supercoiling. So when the hydrogen bonds are breaking down in between the two strands of DNA, then on the opposite side the tension increases. The tension in the DNA helix increases, and that is known as the supercoiling. Okay, that is known as supercoiling. So during the supercoiling process, the topoisomerase, this is the replication fork. Okay, this is Y substructure and this is called replication fork, and above the replication fork this is the coiling twist, this is the twisting or coiling. So enzyme topoisomerase binds to this supercoiling part and it cuts the two strands of DNA. After that, the topoisomerase enzyme cuts the two strands of DNA here. These two strands are being cut down by the topoisomerase. Then next one here, see. So it cuts down both the strands. Then this type of coiling is now relapse. Okay, this type of coiling is now relieved. So this way it will make a complete uncoiled part. That means simply the topoisomerase enzyme cuts the DNA strand and reseal the DNA strand. Okay, so topoisomerase enzyme cuts the two strands of DNA to relieve the stress. This is called stress. The supercoiling is called stress. Okay, the topoisomerase enzyme cuts the two strands of DNA to release the tension or stress. So after cut down of the two strand, it again reseal. Okay, it again reseals the DNA strands. Okay, this way it will help in the formation of one uncoiling part. Yes, so this one is the supercoiling. So topoisomerase enzyme relieves the supercoiling here by remove or by suppose splicing the two strands of DNA. Again it reseals the two strands of DNA, and this way it will form one complete uncoiled or unwinded part of DNA helix. Okay, so this is the function of the enzyme topoisomerase. So now let's back to our previous slide. Here see the last one, single stranded binding protein. So the single stranded binding protein binds to the single stranded DNA. Okay, when the helicase enzyme breaks down the hydrogen between the two strands of DNA, then this separated stranded are hold together by or stabilized by the single stranded binding protein. Okay, after separation of the two strands are separated when the helicase enzyme breaks the two strands of DNA. Okay, when the helicase enzyme breaks the hydrogen between the two strands of the DNA, then the single stranded binding protein binds to the single stranded DNA and stabilize the both strand of DNA during the process of DNA replication. So remember, these are the very important enzymes involved in the DNA replication process in prokaryotes. Okay, DNA polymerase 1, 2, 3. Then helicase enzyme important. Ligase means which join two different segment of DNA. Okay, primase which can produce the RNA primer, sliding clamp. This hold the DNA polymerase in place during the DNA replication. Topoisomerase it cuts and reseal the two strands of DNA, and this way it can relieve the stress or tension in the DNA molecule. Single stranded binding protein it stabilizes the separated strands of DNA. Okay, so these are the important enzymes involved in the process of DNA replication. Okay, now let's move to our next slide. Here, so now the steps involved in DNA replication. So here say first we need the activated deoxyribonucleotides. Okay, because these are the important materials for the synthesis of the new DNA strand. We need ATP, GTP, CTP and TTP. But remember, all these deoxyribonucleotides are found in the nucleoplasm in the form of monophosphate. Okay, remember. So first we need the deoxyribonucleotides for the synthesis of the new DNA strand, and all these deoxyribonucleotides are found in the nucleoplasm in the form of monophosphate. Monophosphate means in the form of deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate and deoxythymidine monophosphate. And these monophosphates are phosphorylated in presence of the enzyme phosphorylase. Okay, and are converted to the active forms which are now called as the triphosphate. That means after this monophosphate are phosphorylated in the presence of the phosphorylase enzyme, now all these monophosphate are converted into triphosphate. That means deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate and deoxythymidine triphosphate. Okay, so first point, activation of deoxyribonucleotides, and all these deoxyribonucleotides are activated by the phosphorylase enzyme. That means when the phosphorylase enzyme adds two phosphate group in the monophosphates, then it will become the triphosphate. And in this form they contain the energy for the production of new DNA strand. That means the DNA replication is an enzymatic reaction which requires a lot of energy, and this energy is provided by this triphosphate. That means 3-phosphate molecule. Okay, so this is the first step involved in the DNA replication, which is the activation of deoxyribonucleotides. So now let's move to the second slide. So next point is origin of replication. That means always we need one specific point from which the replication process will begin. Okay, and that specific point is called as the replication origin or origin of replication. In prokaryotic DNA, remember, in prokaryotic DNA only one point of origin present. That means only one origin of replication present. That means from only one specific point the DNA replication begins. And in E. coli bacteria, this specific point is called as the ORIC. ORI means origin of replication. Okay, ORIC. Remember? So origin of replication means after getting the deoxyribonucleotides, the next step is the recognition of the origin of replication, the point of origin. Okay, the point from which the DNA replication will begin, and that is called as the ORIC. That point is called ORIC in E. coli bacteria. Okay, and ORI meaning origin of replication. Remember, origin of replication means the point from which the DNA replication begins. But in eukaryotic DNA, there are several thousand of origin of replication. Okay? In prokaryotic DNA, only one point of origin present, but in case of eukaryotic DNA, there are several points of origin present from which DNA replication begins. Very important. Okay. So after getting the origin of replication or point of replication, the next one is unwinding of DNA helix. That means separation of two strands of DNA helix. So I have told you already that the enzyme helicase breaks down the hydrogen bonds present between the two strands of DNA. Yes, this is called as the unwinding of DNA helix. So to produce the new DNA strand, the parent strands must be separated from each other. Yes. Then these two separate strands will function as the template strand for the formation of the new strand. So the enzyme helicase, remember all these highlighted names, the helicase one. The enzyme helicase unzips the two strands of DNA. Unzips means unwind. Okay, it opens up the two strands of DNA by breakdown of the hydrogen bond. So enzyme helicase unzips. That is why this is called unzipping enzyme also. So the enzyme helicase unzips or unwind the two strands of DNA by destroying the hydrogen bond present between the two strands of DNA or present between the nitrogenous base pair. Yes. Then the separated strands are stabilized by single-stranded binding protein. I have already told you about the single-stranded binding protein. That means after separation of the two strand, the single-stranded binding protein stabilizes the separated strand. Okay, so after that, say the unwinding of DNA creates tension on the distal part of the DNA molecule, which is released by the enzyme topoisomerase. So topoisomerase cuts and reseals the DNA molecule to release the tension. During the unwinding process of DNA, there are supercoils formed in the winding part or attached part of the DNA helix. So to release that supercoiling or release that tension, the enzyme topoisomerase cuts and reseal the two strands of DNA to avoid that supercoiling or avoid that tension. Okay, so here see, then cause nick means gap, and reseal the DNA strand. So they can cut or reseal the DNA strand to avoid that supercoiling or tension. So unwinding of DNA helix result in the formation of the Y-shaped structure called replication fork. That means when the two strands of DNA are separated from each other due to breakdown of hydrogen bonds between the nitrogenous bases, then the separated strands appear as Y-shaped structure, capital Y type of structure, and this structure is called as the replication fork. Remember, so this is very important question. What is replication fork? Replication fork is the structure of the DNA helix after separation of the two strands of DNA with the breakdown of hydrogen bonds present between the nitrogen base pair. Okay, so now let's move to our fourth point. The fourth point is the formation of the RNA primer. So without RNA primer, the production of the new DNA is impossible. Why? Because RNA primer contains the 5' and 3' direction. The DNA polymerase III enzyme only can polymerize the deoxyribonucleotides to the 3' end of the newly synthesized DNA or newly synthesized strand. That means the DNA polymerase III polymerize or add the deoxyribonucleotides, that means ATP, GTP, CTP and TTP to the 3' end of the RNA primer, and this way it will polymerize or it will continuously add the deoxyribonucleotides to make a long strand. Okay, so here say the RNA primer is a small strand of RNA which is synthesized at the 5' end of the DNA, new DNA strand. Okay, with the help of DNA specific RNA polymerase called the primase. Remember, The RNA primer is a small strand or short strand of RNA which is produced by the primase enzyme, where at the 5' end of the new DNA strand. Okay, so next, the DNA polymerase enzyme binds to the primer. Remember, before production of the new DNA strand, first the DNA polymerase 3 binds to the primer, this RNA primer, and continuously adds deoxyribonucleotides, that means ATP, GTP, CTP, TTP at the 3' hydroxyl group of the new strand. Okay, so once the initiation of the DNA synthesis completed, the RNA primer is removed enzymatically. That means RNA primer is a short strand of RNA which is produced by the primase enzyme. During the process of DNA replication, the enzyme DNA polymerase 3 binds to this RNA primer and continuously add deoxyribonucleotides in a long chain from this RNA primer. Okay, that is why the RNA primer is very important. That means without RNA primer, DNA polymerase cannot synthesize any new DNA strand. Okay, are you getting my point? Remember, RNA primer is very important, and once the initiation of the new DNA synthesis is completed, then this RNA primer is automatically removed. That means this is removed by the enzyme. And which enzyme removes the RNA primer? Do you remember? Yes, which one? Here see. DNA polymerase 1. So DNA polymerase 1 removes the RNA primer. Once the initiation of the RNA strand synthesis is completed, the DNA polymerase 1 enzyme removes the RNA primer from that long strand, and to fill up that gaps, it can synthesize the new DNA sequences. Okay, and these fragments are joined together with the help of ligase enzyme. Okay, now again let's move to our next one. Okay, so formation of RNA primer. So RNA primer is very important, produced by the primase enzyme, and DNA polymerase 3 binds to this RNA primer before production of the new DNA strand. Okay, so here see in this diagram, this is the 5' end, this is the 3' end of the DNA. Okay, where this one is the newly synthesized DNA strand, where this part is, here see, this part is RNA primer. Yes, this dot part, these are the RNA primer. Okay, the first one, this one is the DNA strand. Okay, and this one, the red one, this one is the newly synthesized DNA strand. This is the template, and this is the newly synthesized DNA strand, where this is the RNA primer, this is the RNA primer. So DNA polymerase binds to the RNA primer and it polymerizes the deoxyribonucleotides one by one. Okay, this way it will elongate the chain of DNA. Okay, so this is the diagram where you can see clearly the position of the RNA primer and DNA polymerase. Okay, so next, see here the last point, not the last, this is the synthesis of the new DNA strand. So after the production of the RNA primer, now the synthesis of the new DNA strand. So DNA polymerase progressively adds deoxyribonucleotides to the 3' hydroxyl group of the newly synthesized strand. Remember, always DNA polymerase 3 polymerize the nucleotide sequences to the 3' hydroxyl end of the DNA. Okay, that means it binds the nucleotides only to the 3' at end, not to the 5' end, and that is why new strand always proceeds from 5' to 3' direction. Why? Because all the nucleotides are added to the 3'. Okay, that is why the chain always proceeds from 5 to 3 prime direction. Okay, so in case of prokaryotes, here see, prokaryotes have three types of DNA polymerase. Yes, DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Remember, in prokaryotes, the DNA polymerase 3 polymerize the nucleotide sequences to produce the new DNA strand. Okay, where the DNA polymerase 1 removes the RNA primer, DNA polymerase 2 it functions as the repair enzyme, and DNA polymerase 3 is the main enzyme which add the deoxyribonucleotide triphosphate one by one to the 3 prime end of the new DNA to produce a long new DNA strand. Okay, so in case of prokaryotes there are 3 DNA polymerase, but in case of eukaryotic DNA synthesis there are 5 types of DNA polymerase, and these are DNA polymerase alpha, beta, gamma, delta, and epsilon. Okay, in case of the eukaryotic cell there are 5 types of DNA polymerase alpha, beta, gamma, delta, and epsilon. Remember, in case of prokaryote only 3, 1 to 3, but in case of eukaryote there are 5 DNA polymerases. So next, here say DNA polymerase 3 synthesize new DNA in 5 prime to 3 prime direction, and the strand with 3 prime to 5 prime polarity serve as template for continuous DNA synthesis called leading strand. That means, remember, if the parent DNA strand is 3 prime to 5 prime direction, okay, then in this strand one new DNA strand will be formed, okay, which direction will be 5 prime to 3 prime direction, and this strand will be known as the leading strand. I will show you in the diagram which one is the leading, which one is lagging. This is very important. The another new strand is called as the lagging strand. That means from one DNA molecule, 2 new DNA molecules are produced. In this 2 new DNA molecule, one new strand is continuous, which is called as the leading strand, and one new DNA strand is discontinuous, which is called as the lagging strand. In the lagging strand, there are some short fragments of DNA present which are known as the Okazaki fragments. Remember, in the lagging strand, lagging means which is not continuous. Not continuous meaning which have some DNA sequences, and these sequences are known as the Okazaki fragments. Okay, and all these Okazaki fragments are joined together by the ligase enzyme. Okay, so the newly synthesized DNAs are called as the leading strand and lagging strand. That means 2 new DNA strands are formed from 2 template DNA strands. So one DNA strand will be continuous called leading, one DNA strand will be discontinuous which will be known as the lagging strand. So now which one is known as the leading and which one is known as the lagging strand? That means from which template the leading strand is formed and which template the lagging strand is formed. Now here say in the diagram, in this diagram this is the 5 prime to 3 prime direction, this one 5 prime here, okay, here see this one 5 prime to 3 prime. This one is the template DNA strand. You know what is template? Template means the parent DNA strand. Okay, so this one is the 5 prime and 3 prime. Okay, 3 prime, this is the parent DNA strand. Okay, so now here say this is the RNA primer which is formed in the 5 prime end of the new DNA strand. This one is already present, which is called the template DNA strand, that is already present, and its direction is 3 prime to 5 prime. Okay, its direction is 3 prime to 5 prime, 3 prime to 5 prime. So here say, from this template strand one new DNA strand will be synthesized. Yes, and this is the RNA primer which is formed in the 5 prime end, and in this RNA primer the DNA polymerase bind. Yes, now when the DNA polymerase binds to RNA primer, it will polymerize, or it will add the deoxyribonucleotides one by one, and this way the chain continues in this direction. Okay, this way the chain continues in this direction. Okay, I will show you again. Here see, this one, you can see direction, you can see the direction, this one. Yes, this is the template strand. Okay, the 3 prime to 5 prime, this is the template strand. This is the template strand where this one is the new strand. Okay, so in this new strand, first the RNA primer is formed. Always in the newly synthesized strand, the RNA primer is formed to the 5 prime end, and to the RNA primer the DNA polymerase binds, and progressively it polymerizes the nucleotide sequences one by one, and this way the chain elongates in this direction. Okay, in this direction the chain elongates in this direction. That means always the new DNA strand is formed from 5 prime to 3 prime direction, and you can see here, this is the 5 prime of the new DNA strand, this is the 3 prime of the new DNA strand. That means the chain will elongate in this direction. Okay, the chain will elongate in this diagram. Okay, so this is called leading strand. Okay, this one is called now leading strand. Which one? This new DNA strand is called leading strand. Why? Because in this new DNA strand there are no gaps in between the DNA sequences. So DNA polymerase will progressively add the deoxyribonucleotides in a long chain. Okay, now see which one is called lagging strand. So this one is called lagging strand, where these are, where this is one, this is one, this is one, these are the fragments of DNA. Okay, so here see, this is the parent DNA strand. Okay, this is the parent DNA strand, and this is the new DNA strand. This is the new DNA strand. Okay, this is the parent, this is the new DNA strand. So in this new DNA strand, here see, the parent strand is 5 prime to 3 prime, 5 prime to 3 prime. Okay, and new DNA strand is 3 prime to 5 prime. Okay, when the parent DNA strand starts from 5 prime to 3 prime direction, then the newly synthesized DNA strand will become the lagging strand. Lagging means which have lags, nicks or gaps. So all in this case the RNA primer is formed. Okay, when the hydrogen bonds breaks down, then every time RNA primer is formed and DNA polymerase binds to the RNA primer, and it will polymerize the short sequence of nucleotide. Okay, again when hydrogen bond breaks down, again one new RNA primer is formed. This way many RNA primers are formed, and DNA polymerase 3 polymerize only few nitrogenous bases or sequences of DNA, and this way many gaps are formed in between the strands of DNA. Okay, that means many short fragments of DNA are formed, and all these short fragments of DNA are filled up by the ligase enzyme, and all these primers are removed by DNA polymerase 1 enzyme. Okay, let me simply remember which one is the leading strand, which one is the lagging strand. Okay, if the parent DNA strand is 3 prime to 5 prime direction, then the newly synthesized strand will be known as leading strand, because the new strand begins from 5' to 3' direction. Okay, only when the parent strand is 3' to 5' direction, then the new strand will start or proceed from 5' to 3' direction, and this will be known as the leading strand. Okay, and on the other hand, if the parent strand contain the 5' to 3' strand, okay, the parent strand is 5' to 3', then the new strand will be in the 3' to 5' direction. Okay, and this strand will be lagging strand. That means many short fragments of DNA are formed in this strand, okay, which are called the lagging strand. Okay, so these are the differences between leading and lagging strand. Just remember from which strand of the parent DNA molecule the leading strand is formed, and from which parent DNA strand or template DNA strand the lagging strand is formed. I will show you again. Here see which are the Okazaki fragment. In the lagging strand, all the here see in the lagging strand, all the short segments of DNA are called Okazaki fragments. So up to here, this is one Okazaki fragment, and this is another Okazaki fragment. This way, many Okazaki fragments may present in this newly synthesized DNA strand. Okay. Remember, Okazaki fragments means the short fragments of DNA which are synthesized in the template which proceeds from 5' to 3' direction. Okay, this is the parent strand which directs from 5' to 3'. Only in this template strand the lagging strand is formed, and in the lagging strand all these are called as the Okazaki fragments. That means these short fragments along with the RNA primer, this is called as the Okazaki fragment. Okay, remember this is very important question for your exam, which is Okazaki fragments, or what do you mean by Okazaki fragments? Okazaki fragments are short segments of DNA which are formed in the lagging strand. Okay, now here see the last point, elongation of the new strand. Elongation of new strand means how the nucleotides are joined together. Very simple. So the DNA polymerase 1 degrades the RNA primer, and it catalyzes the synthesis of the short DNA segments to replace the RNA primer. Yes, so this way these short segments are joined together by the ligase enzyme, and this way the long chain of the DNA is formed. Okay, that means here see, okay, you can see here, okay, here see in this diagram. So in the lagging strand, these are called as the Okazaki fragments. Yes, these are Okazaki fragments. So these Okazaki fragments, all the Okazaki fragments contain this primer. These are called primer. Yes, RNA primer. So when the new Okazaki fragment is forming, then the old RNA primer present in the old Okazaki fragments are removed by the DNA polymerase 1. Okay, along with the formation of the new Okazaki fragments in the lagging strand, the old RNA primers are removed by DNA polymerase 1, and to fill up these blanks, the DNA polymerase 1 can synthesize the short nucleotide sequence of DNA, and this short nucleotide sequence of DNA are joined together with the help of the ligase enzyme. Okay, so that is all about the elongation of the new strand. This way the RNA primers are removed, and in those places the new DNA strands are formed by RNA polymerase 1, and all the gaps are filled up by the ligase enzyme. And this way RNA polymerase 3 will polymerize the nucleotide sequence one by one to produce a long strand of DNA. Okay, here see in this diagram. So this is Okazaki fragment, this is Okazaki fragment. So these are two Okazaki fragments, but you can see there are no RNA primer here. Okay, so this RNA primer is already removed by the DNA polymerase 1, and in those places these nucleotide sequences are formed. Okay, after that the gaps between these two Okazaki fragments are filled up by this ligase enzyme. Okay, the yellow one is the ligase enzyme which joins the two strands of DNA. So this way the long chain of the DNA elongates. So one by one the RNA primers are removed by the DNA polymerase 1, and in those places the new sequence of DNA are formed, and all the short segments of DNA are joined together by this ligase enzyme. Okay. Proofreading and DNA repair. So proofreading means sometimes during replication wrong base pair may be introduced. Okay, this is very rare, but sometimes may a wrong base pair may be introduced. That means sometimes along with the A guanine binds. Okay, this is the wrong base pair because adenine always binds to thymine. Yes, so in this case the DNA polymerase 2 can correct this wrong base pair. That means it can remove the wrong base pair and it can join the correct base pair in those places. Okay, this is called as the proofreading or DNA repair. And in proofreading or DNA repair, always the DNA polymerase 2 is important. Remember this thing. DNA polymerase 1 remove the RNA primer. DNA polymerase 2 works as or serve as the repairing process which can remove the wrong base pair, and DNA polymerase 3 is the main enzyme in prokaryotes which polymerize the new strand or new DNA strand. Okay, now let's move to the next one. So this is the complete diagrammatic representation of the replication, of the mechanism of DNA replication. Okay, so you can see here this is the double stranded DNA. Okay, in this double stranded DNA, this is the topoisomerase. This one, the green ring, is the topoisomerase which relieves the tension or which relieves the supercoiling. Okay, above the replication fork. This is called, this whole part is called as the replication fork. Okay, these two are, this part is called as the replication fork, which is the Y-shaped structure. Okay, so now here say, so topoisomerase, now all these purple color are single stranded protein. Okay, which binds to the single strand of the separated DNA strand. Here also these are the single stranded binded protein which stabilizes the two strands of DNA after separation. Okay, now this is the RNA primer which is very important. RNA primer recognizes the initiation or origin of replication from which the replication process begins. Okay, so here say this is the primase enzyme. The ring, the blue ring, is the primase enzyme which helps in the production of RNA primer. Then the helicase enzyme, this one is the helicase enzyme which continuously breaking the hydrogen bonds present between the two strands of DNA, and along with the breakdown of the hydrogen bonds these two strands are separated from each other. Okay, so this is one strand, this one and this one, these are the parent strand or template strand, where this one template strand starts from the 5 prime to 3 prime direction, and here this template strand starts from 3 prime to 5 prime direction. And you can see here the template strand which is directed from the 3 prime to 5 prime. In that template strand, the new synthesized or newly synthesized DNA strand is from 5 prime to 3 prime direction. That means this new strand is moving inwards. Okay, this new strand is moving inwards, which is from 5 to 3 prime direction, and this will be known as the leading strand. There are no gaps in this strand. Okay, on the other hand, in the opposite strand, where the parent strand is from the 5 prime to 3 prime direction, in this case the newly synthesized strand moves from inside to outside. In this case the new strand is moving from inside to outside. Okay, but in the leading strand, in case of leading strand, the leading strand always move inward, inside, and the lagging strand move outside. And in this strand always some fragments of DNA are formed. Okay, which are called as the Okazaki fragment. All these are fragment, all these are gaps, that means all these are fragment of DNA, and all these fragments are joined together by the ligase enzyme. Okay, remember If the parent strand is from 3' to 5' direction, the new strands will be the leading strand, which will be from 5' to 3' direction. On the other hand, if the parent strand is 5' to 3' direction, then the newly synthesized strand will be lagging strand, which will be from 3' to 5' direction, or you can say that this strand will continue from inside to outside of the replication fork. Okay, and the leading strand always move inward of this replication fork. So this is the complete mechanism of DNA replication. Okay, so if you have any doubt, you can ask me. So in this process, you just have to remember the functions of different enzymes, and very important point, what is leading strand and what is lagging strand and what is Okazaki fragment. So remember all these things. Okay guys, so thank you.